There we go. Woo. All right. Yeah, so if you have to leave early or if you have, you know, friends or whatever that couldn't make it. Here's another person coming. Cool. Okay. So yeah, so typically I just have you guys run it with your questions, and it ends up being a really good review of the topics on the exam. I finished making it last night, so it's up. So we did energy, enzymes, as well as cellular respiration and cell division. That's this unit's unit 3. So who has questions? I'll pull up the chat. Perfect. It's so funny. So funny. That's the best question you could ask, because that was exactly the very first question of my review session for my other school was the same thing. Can you go over the Kreb cycle? So yes, that's a great question. Let's do that. How shall I begin? So the best way to begin this discussion is to do a brief summary of what happened before the Kreb cycle. And so this is, right, cellular respiration. Let this person in. Cellular respiration, this is the catabolic process by which cells are oxidizing glucose in a series of chemical reactions occurring one after the other in these redox reactions, so-called redox, where electrons and hydrogen are being stripped off the molecule in that particular reaction. And it's a sequence of these reactions. The first phase of these reactions is called glycolysis, right? And glycolysis, right, that was when we started with glucose and we ended up with two, who remembers the two molecules are end products of the glycolytic pathway? What were those two molecules called? We did get NADH, we did get ATP. Yes, so those are considered byproducts of the reactions. Absolutely. No FAD, but pyruvate or pyruvic acid, same thing. Pyruvate, pyruvic acid. So pyruvate. So that's where we left off in glycolysis, right? Remember, this is kind of I always like to think of this whole thing as an assembly line in a factory. And we've got this starting thing, glucose, that we're continually modifying in enzyme catalyzed steps, enzyme catalyzed reactions. And as we're modifying, we end up with two now, right? Glycolysis. We split the sugar into two pyruvate molecules. So you could say two pyruvates, two of them, right? And recall that glucose, we start with a 6-carbon molecule. Glucose has 6 carbons. And so at the pyruvates, because we split glucose in half in glycolysis, each pyruvate has 3 carbons in it. So we have 2 3-carbon molecules called pyruvate, 2 pyruvate molecules. Or pyruvic acid, depending on the conditions, it's same thing. Okay, so I start there because that is really the first, going along with this assembly line. The 2 pyruvates are then going to enter another part of the factory in this analogy, being the mitochondria. So before we do the Krebs cycle, remember, we're entering stage 2 now. Stage 1 is glycolysis. And you get, as some of you mentioned, we get 2 NADH molecules. We also get 2 ATPs out of glycolysis. So we've already got a little bit of energy. Cool. It's not nearly enough for us, a multicellular oxygen breathing organism. We need a lot more. And so we got to keep doing things here. And so we've got 2 NADHs, great. We've got 2 ATPs, awesome. 2 pyruvates. All right, let's continue oxidizing pyruvate. So remember, stage 2, doing red, consists of 2 phases within it. There's 2A, I call it, which is pyruvate oxidation. Try and get my light on here too. There we go. Pyruvate oxidation, right? And pyruvate oxidation had a few names. If you're reading the text, it's typically called that, but it's also called pyruvate processing or the transition step or whatever. It's all the same, but I like to call it pyruvate oxidation because that's what's going to happen. Now, keeping in the back of your mind, we have 2 pyruvates from glycolysis, right? We have 2. So for this example, I'm just going to go through it with 1 because that's the process. It just happens 1 molecule at a time. But at the end of all this, remember, we're going to double everything because we have 2 pyruvate molecules, right? But let's just start with 1 and go through the process with 1 of them. So pyruvate oxidation, here's our pyruvate that we start with, right? One of them I took from up here. Just here we go. So pyruvate oxidation is 2 or 3 steps. Not too important, but I wonder if I can remember from memory the exact numbers, but let me see. Okay. During pyruvate oxidation, there's several steps, like 2 or 3. You don't have to know that. But remember how in the lecture I drew a little more detail on the pyruvate molecule and I added this carboxyl group at the end of it, which is fancy for a C with 2 oxygens, carbon with 2 oxygens bound to the molecule. And I do this because one of these steps in pyruvate oxidation is a decarboxylation reaction, whereby an enzyme, a decarboxylase enzyme, is going to cleave that bond. It's going to break it off. And so in one of these pyruvate oxidation steps, we have the release of what molecule? What is that with C with the 2? I just showed you, but I covered it. Ha. What are we releasing? Carbon dioxide, CO2. Exactly. It's a C, right? A C. I know, amazing, right? C with 2 O's is coming off. So if I were to ask you on the exam, wink, wink, when the first molecule of carbon dioxide is released during cellular respiration, when would that be? what we're talking about, right? Correct, pyruvate oxidation. Pyruvate oxidation. That's when the first, right? We just saw it. We don't get any CO2 from glycolysis. The first one comes from pyruvate oxidation. Okay, what else comes from pyruvate oxidation? We get an oxidation, right? Hence the name. That's why I like this name. It helps you remember, oh, we're also going to generate an NADH molecule, like we did in glycolysis. So let me draw it out here. Okay, so we got a CO2, but we also have an oxidation take place, meaning there's another enzyme, pyruvate dehydrogenase, that helps catalyze the oxidation of pyruvate, whereby, as you can see, 2 electrons and a hydrogen are stripped off pyruvate in one of these reactions and stuck onto NAD+. And when that happens, we get, of course, NADH. So we've remembered what oxidation is. Oxidation, by definition, is when you remove electrons from a molecule and hydrogen. In biology, it's always associated with hydrogens as well. So if you see a hydrogen and electron, or electrons in this case, stripped off of a molecule, taken away, that's oxidizing that molecule. Now, what if I asked you, NAD+, was NAD+ reduced or oxidized? Am I adding electrons and hydrogen to NAD+ or removing them? Yeah, remember, it's called redox for a reason. Whenever you oxidize something, take away electrons and hydrogen, well, there's going to be another molecule that accepts them, gains them. And so in this case, NAD+ is reduced to NADH because we added electrons and hydrogen from pyruvate. I'm kind of going over this because I'm pretty sure I asked a couple questions just on the principles of reduction oxidation. Okay, so at the end of pyruvate oxidation, oops, there you go. The final product is a molecule called, oh, wait, so go to the chat. Oh, this is hard. There we go. Got it. Oh, 2 questions here. Beautiful question. Both of them. So Rachel, yes, if you reduce a molecule, you are adding electrons to it. Why? Because, well, reducing, think of the charge. Electrons are negatively charged. So let's say this pen is an electron, okay? And here I am, and I accept this electron from another molecule, I gain it. Well, I've just been reduced. My overall charge has gone down, reduction, because electrons are negatively charged. Oxidation, yeah, oxidation is the opposite. If someone came over and took this from me, stripped it away with hydrogen electrons, taking them, I have now been oxidized. Pyruvate oxidized and NADH is reduced. So Jody, to be more accurate, because that's close, pyruvate is oxidized, but it's NAD+ that's reduced to form NADH. But I think I know what you mean. So just to be, so like the concept is that this, where does the NAD+ come from? Vitamins that we eat or get from our food. You know how like vegetables, fruits, they have this vitamin in their cells because they do the same thing, actually, the plant cells. So we get it from them. organic compounds known as vitamins. They're coenzymes, right? Which is part of the enzymes lecture. We talked about how there are certain enzymes that need helper molecules, such as NAD+, to catalyze this oxidation. No worries. So what does the 2E and H+ mean? So let me show you. So the 2E minus, where is it? 2E minus, 2 electrons. So what's happening here is that NAD+ is coming in during this process and assisting an enzyme to strip off 2 electrons and a hydrogen ion from pyruvate, or one of the intermediates in the pathway. And these particles, the electrons and the hydrogen ion, are going to be stuck, attached to NAD+, and then it pops off and you get an NADH. Pops off from the enzyme. Yeah. Okay, cool. So while I go get some water, does anyone remember the final ending molecule of pyruvate oxidation? It started with an A. Right. Yes, acetyl-CoA. That's right. Okay. So acetyl-CoA, an acetyl group, has 2 carbons in it, and it's attached to this coenzyme A, CoA. So this is oops. Okay, so at the end of this process, pyruvate oxidation, you're going to get this 2-carbon molecule attached to this coenzyme, right, called acetyl-CoA. Now it's 2 carbons, right, because we lost one as CO2. So we end up with acetyl-CoA. Okay. Now, I'm gonna go ahead and keep this, right? I'm not gonna like get rid of it because we're gonna come back to it. Because remember, we're doing all of this twice, so I'm gonna double everything at the end. But for now, let's just keep going with the overall sequence. Do the 2 carbons equal acetyl-CoA? Oops. Or bond with it? No, the 2 carbons are acetyl-CoA. Yeah. So that I wrote acetyl-CoA because that's what this is. 2 carbons and the coenzyme A. Coenzyme A is bound to these 2 carbons through a bond. It's a giant molecule, though, so we just write CoA. It's another coenzyme like NAD plus, that is, or FAD. Just another coenzyme that you don't have to know why it attaches and helps out here, but it's necessary for the next step. Let's just say that. And so again, as we go through our assembly line, acetyl-CoA is the molecule that enters the Krebs cycle. Did you say CoA is bound to the 2 carbons? Yes. Yeah. You've got 2 carbons covalently bound together, and then attached to one of them is coenzyme A by another covalent bond. I just do the squiggly line just kind of to represent that CoA is a giant molecule. But yeah. So now we enter, so fancy. Now we enter part B of stage 2, which is the Kreb cycle or citric acid cycle, same thing. So Kreb cycle we're going to enter now. Okay, so we left off with acetyl-CoA, right? And so from pyruvate oxidation, the acetyl-CoA comes over. So I'll draw that first. And it's going to enter this cycle. So here it is. It will enter the cycle. And it does so by joining, linking up, attaching to the first molecule of the Kreb cycle, the starting molecule, which is, it has 4 carbons. Okay, so it enters this cycle. Remember, it's a cycle that actually is going to end up regenerating this starting molecule. Okay, so acetyl-CoA joins into the cycle by attaching to this 4-carbon molecule. And it's called oxaloacetate. It's the very first substrate or molecule of the Kreb cycle. And so that's really the first step of the Kreb cycle, is acetyl-CoA from pyruvate oxidation linking up with an oxaloacetate molecule. Okay. How should I do this? Okay. Okay, so that's the first step of the cycle. And remember, there's going to be like 8 total. And so because acetyl-CoA has 2 carbons, oxaloacetate has 4, the first product of this pathway is going to be a 6-carbon molecule called citrate. By this time, the CoA leaves. The bond to CoA breaks off. So CoA has done its job. It catalyzes this first reaction. It helps, at least, because it requires a lot of energy. CoA is high energy, but you don't have to know that. All you have to know is that first step, when the CoA bails, the 2 carbons join to the 4-carbon compound here, oxaloacetate, to form citrate. So this is citrate, and it has 6 carbons. That's our very first step. And again, so this is intro bio, so we don't go into all the 8 steps. We just do the broad overview. And so within these 8 steps, or 7-ish more steps, there's going to be decarboxylations, where CO2 is released. There's going to be oxidations, where you get NADH, as well as FADH2, the other electron carrier and molecule that's reduced. And then you also get a little bit of ATP as well from the Kreb cycle, just like glycolysis we did. And so I'm going to kind of represent all these steps as little arrows going in the cycle. So I'll show you what I mean. So they're all, again, enzyme catalyzed steps occurring one after the other. There's going to be oxidation steps. There's going to be steps where CO2 is broken off of the initial citrate. And that makes sense, right? Because in the end, it's a cycle. So we regenerate our starting molecule. We regenerate the oxaloacetate so that it can do the cycle again. And oxaloacetate, again, has 4 carbons. So we got to lose 2 from citrate, right? In order to get back. So I sort of, let's do that, do it in green. Okay, check it out. So I just cleaved off, I cleaved these 2 covalent bonds, I broke them. We cleave the bond. And so those carbons with 2 oxygens that are bound to them, that I didn't draw that in, but they're there. In 2 of these steps, we're going to get a CO2. So we end up getting 2 CO2s in the one turn of the cycle, right? Okay, that's the first thing we get. Keep that for a second. In a few other steps, we're also going to get, there's actually going to be 3 oxidations of any of the substrate, whether it's fumarate or malate or whatever is in the next intermediate in this pathway. And so we're going to get 3 NADHs out of one turn of the Krebs cycle. So we get 3 NADHs. So 3 NAD pluses are reduced to NADH. We get 1 ATP molecule. One of the steps of the Krebs cycle, you get an ATP. It's one of the 3rd or 4th down here. So you get 1 little ATP. But yeah, just keep it in that. So you get an ATP. And then the last, of course, the other electron carriers involved in the Krebs cycle alone. It's not, so far we've only been dealing with NADH, right? And the oxidation of the substrate to generate NADH, but now we're gonna get from FAD, doesn't have a charge, FAD, we're gonna get 1 molecule of FADH2. And if you're wondering, if you're an in-depth thinker and you're wondering like, why now? Why only this do we deal with FADH2? It's a lower energy molecule. Yes, that's correct. You only get 1 FADH2 for the turn of the cycle, 1 cycle. Is that legible? You can see that, right? Just get 1 of those FADH2s. It's a lower energy molecule, so the oxidation step, if you were to try it with NAD, it doesn't release enough energy to stick electrons and hydrogen onto NAD+. So a different lower energy molecule is required. We'll just keep it at that. It gets complex biochemistry stuff there. Okay, but that's it. Now, the only thing left that we have to do, and keep in mind, is that per molecule of glucose that we start with, we end up with 2 pyruvates, right? Before the 2nd stage. So we have to double everything, because we're going to do it all twice. I only showed you how we do it with 1 pyruvate we started with. So we're going to do everything twice. So all the products we're accumulating, all the molecules, are going to be doubled. And so maybe I could do that in like a different color that I've not used yet. Let's do blue. So pyruvate oxidation, right? You're actually going to have two pyruvates, right? Well, I kind of already wrote that up here. But we're going to do that process twice because you have two pyruvates. So you get two NADHs total from pyruvate oxidation. You get two CO2s, right? Two CO2s. Let's circle it. Make it. There you go. You get two acetyl-CoAs. Two acetyl-CoAs. Great. I know there's a trick question on there, so on the exam. So how many ATPs did I produce in pyruvate oxidation? How many ATPs? Not two. 0. Yeah. So I didn't write ATP on here, did I? No, I've only made CO2 and NADHs. Yeah, no ATPs here. So if I asked you how many ATPs are made in pyruvate oxidation, you'd probably say 0, right? Because none are made. I'm a tricky boy. Let's see. But glycolysis makes 2, right? Absolutely. Yes, that's correct. And so does the Krebs cycle. Well, per turn, there's 1 ATP, right? So I'm still doubling everything because since we ended up with 2 acetyl-CoAs, we do the Krebs cycle twice. 1 turn per acetyl-CoA, and we have 2. So we're gonna have to double everything here too. So I'm gonna double the CO2s. So there's 4 CO2s total from the Krebs cycle. I'm gonna double the NADHs, so 6 total. 2 ATPs and 2 FADH2s. So that looks like that. Okay, that's it. As far as the Krebs pyruvate oxidation goes. So questions about that? I know. Isn't it great? Oh, good. Yeah. I agree. It's this unit especially is one that you kind of have to, revisit. Because it's a lot information one after the other, So Krebs cycle produces 2 ATPs, yes. And pyruvate oxidation produces 0. That's absolutely correct. Yeah, no ATPs from pyruvate oxidation, but Krebs, yeah, you get 2. So yeah, so glycolysis and Krebs and all of stage 2 together, you get 4 total ATPs, which is again not a lot. So it's really the majority of ATP that you get from the electron transport chain. Oops, dang it, it's considered The intermediate step. Oh, there's no intermediate step in the Krebs cycle. If I mentioned an intermediate, that's just referring to one of the molecules in the Krebs cycle that, remember, it's a series of chemical reactions that occur one after the other. So it'll be reactant product. Oh, then it's the pyruvate. Okay, so it's pyruvate oxidation. Yeah, it has a bunch of names. Transition step, intermediate step. Yeah, sorry. I guess I call it that in the study guide. There are study questions. So other questions, I think that was pretty good review for that. There's a few questions about sort of, how many ATPs from Krebs or whatever, how many, whatever from whatever. So I think so, at least. I made it last night, but I think I did a few. So that'll be helpful. Other questions, we did that's a pretty good review of cellular respiration. There's also electron transport chain, there's cell division. Okay, let's take a look. Why is the total ATP production different in prokaryotes and eukaryotes? Did I cross that out in the study questions? I should have crossed that out because I don't think I talked about that in lecture. I did cross it out? Okay. Yeah, so yeah, I can still answer it. It's very quick. Essentially, eukaryotes, that's kind of a good review of understanding. So remember, glycolysis takes place in the cytoplasm, right? And then so if you remember glycolysis, you get 2 NADHs, 2 ATPs, and really that's it. And the 2, of course, pyruvates by the end. But because it's in the cytoplasm, the NADHs have to go to the electron transport chain eventually. And it actually requires active transport. So eukaryotes have mitochondria to shuttle them in with energy. And so prokaryotes do not. Remember, they once were the mitochondria. They do this in their plasma membrane, the electron transport chain. So there's no energy expenditure to get NADH there. It's just kind of floats on over in the cytoplasm. But you don't have to know that. Yeah. Thank you, Rita. Yeah, no problem. Other questions? Oh, great question. So we talked about the cell cycle and the various cell cycle checkpoints where the cell is going to check some things before proceeding in the cycle. And so let me draw the cell cycle really quick. Okay. Here's my cell cycle. So the cell cycle, we start with G1, then S phase is after G1, then G2, and then M for mitosis, cell division. There are a couple checkpoints that the cell has to really pass. Can't fail these checkpoints in order for the cycle to keep going and to proceed. Like there's a G1 checkpoint, there's a G2 checkpoint before cell division, and then there's also a checkpoint in mitosis, right? That metaphase checkpoint. So I kind of drew some lines here. And really these checkpoints are checking for, it's here, it's, yeah, just the G1 checkpoint. The cell will check for proper nutrients available in the surrounding environment. It needs nutrients to grow, so it won't keep growing unless there are nutrients around. But that question in particular is referring to the G2 checkpoint. And essentially, when a cell fails the checkpoint, G2, by the way, who remembers what G2 checks for? At the end of G2. Abnormalities, what kind of abnormalities? More specifically, what kinds of mistakes? There it is. Yes, the cell checks for accurate DNA replication. So when does DNA replication occur? It occurs in S phase, right? That's when all of the DNA is copied, right? We have 3.2 billion nucleotides of DNA. So it's a lot of copying. And it's a lot of errors that could happen, right? Well, the enzymes that copy DNA, which we'll learn about more in the next unit, they're remarkably accurate. There's a mistake every 1 in 1,000, roughly. And essentially these mistakes, these in DNA replication, these errors can be corrected by other enzymes. There's proofreading enzymes, repair enzymes that'll fix the mistakes. So by the time it gets to that checkpoint, should be good to go. The cell will scan it and be like, okay, everything's good. Well, sometimes it's not, especially in cancerous cells, or precancerous, that is. So let's say it fails that checkpoint and the mistakes are too large to correct with these proofreading enzymes. Just too much error, too many nucleotides that are wrong. The cell has two options, right? It can repair, but if it can't, the damage is too extensive, like a ton of nucleotides are wrong, the cell will actually undergo programmed cell death, which the word for that is called apoptosis, which is a very organized, controlled way to recycle and cell components and degrade them. And other cells actually come in and eat it up and use their components for their own nutrients or whatever. It is creepy. I agree. But it's really interesting. Apoptosis, the mechanism, has led to a lot of amazing research and studies. Apoptosis is actually the reason you have fingers, if you didn't know that. We're all born in the embryo, we have webbed hands. Our hands are webbed together. It's one big web being like ducks have, you know? But apoptosis genes are turned on in between. And so the cells that comprise in between your fingers and the webbed embryo fingers actually kill themselves, program cell death, and so it leaves the spaces in between. So apoptosis is a really useful mechanism for a lot of different things. That's just one in development, but yeah, there are a lot of preventing cancer too, cancer cells that need to die. So I think there's another really huge advantage of apoptosis is when you're developing your immune cells in the thymus or the bone marrow. So your white blood cells that fight infection. Well, you don't want those cells to attack yourself, right? You don't want your white blood cells to release a bunch of like super destructive, gnarly enzymes against your own skin or your liver or your heart or whatever that can kill you. It's autoimmune disease. It's bad. So cells that are self-destructing, autoimmune cells, so there's a programmed cell death for those types of cells when they develop. So it could be useful. But yeah. Other questions? That's a great question. Oh, yeah. You got it. I think this is probably one of the toughest units. Well, I say that, but then the next unit, I hate saying that because then I have students that are like, I thought you said the last unit was hard. Well, it's funny because it's like some people think this is their best, and they think the previous unit was harder, and it just varies. People have strengths and weaknesses. I think my toughest personal unit in biology was respiration. It took me a long time to remember that kind of stuff. But it happens. Just a lot of studying, a lot of practice. Any questions on cell division? Mitosis, meiosis. Independent assortment, yeah, it's a good question. So when does independent assortment occur? What phase of meiosis? Not metaphase 2, metaphase 1, right? Let me draw a cell here. Okay. This is metaphase 1. And my example here has 4 chromosomes, 2 homologous pairs. Oh, metaphase 1, there it is. Oops. Okay, there you go. All right. So independent assortment, all it means is the way I drew these chromosomes lined up like this one next to this one. So these might be your 2 chromosome 1s, 2 chromosome, I don't know, 20s because they're smaller. Remember, we have 23 pairs that line up. Just the way I drew them, on which side each one is. I could have done it like this. Every pair lines up independently, randomly. And we call that independent assortment. So this is what I did. I just switched these 2 places. And now they're down here. That's the after I switched them, right? I could have easily just drawn it that way. And that's 2 different options, right? So when I split this cell down the middle after the end of meiosis 1, I'm going to have different combinations, different variations genetically. The different combinations of chromosomes, the outcomes of chromosomes. And so independent assortment is this way that you can get a huge amount of genetic diversity. A massive amount of unique genetic combinations in your sperm and egg cells from meiosis. Keeping in mind that each pair lines up randomly with whatever side. There's no known, like, it's just totally random how they line up. Each homologous pair. So if you have 23 pairs, the math comes out to 23 to the second, no, sorry, 2 to the 23rd power, which is 2 times 2 times 2 times 2 times 2 times 2, 23 times, right? And it's like roughly 8 million possibilities. So that's what independent assortment refers to. How about crossing over? When does crossing over occur? There it is. Jeannie, you answered my question before I asked it. Amazing. Yes, crossing over occurs in prophase 1. And yes, in this example, I didn't include any crossing over. But, you know, I could have had a crossing over event occur between these 2 and put a red here and a blue here, and parts of the non-sister chromatids, right, swap chromosome fragments, right, or whatever, segments. Yeah, and again, crossing over is another way that this immense genetic diversity can occur, because when you do that crossing over, well, now that chromosome has part of the paternal homolog and mostly the maternal or whatever, and it's an entirely new thing, and there's a new set of genes that just came over. So it creates yet another possibility. So the 8 million combinations doesn't even include that. So that's why it's like astronomically impossible for two siblings that aren't identical twins, because that's an entirely different thing, to be identical genetically, or even that similar. I mean, there are obviously cases because there's so many humans, but not identical. It's like nearly impossible. Can you repeat what I said about not? Yeah. So let me use a green here. All right, hold on a second. Okay, so you see how I pointed up here to these two chromatids on these separate chromosomes? They're non-sister chromatids, right? This chromosome here, the paternal chromosome, has two sister chromatids attached to one another, right? So does this maternal chromosome. But the two chromatids next to each other are non-sister chromatids. So that's the only time crossing over occurs, it's between non-sister chromatids. Because if crossing over happened between sister chromatids, they're identical, right? So it wouldn't do anything, really. Crossing over might occur between sisters. I don't even know, but it wouldn't matter because it's the same, same information. These are different though, right? Similar but different, right? Homologous pairs. Same gene, different alleles or versions of that gene. This was the whole, we both have the same gene for hair color. So hair color gene, same gene, but it could be a different version. It could be a blonde version or a brown or a red or whatever, but it's a hair color gene. Same thing with eye color and yeah. Other questions? No problem. Yeah. Can you go over diploid and haploid? Yes, absolutely. Okay. Okay, so diploid strictly refers to the fact or the situation where a cell has two sets of chromosomes. Actually, so diploid means two sets of chromosomes. So we are diploid organisms. Really, any organism that is the result of sexual reproduction will be diploid because you get a set of chromosomes from one parent and then another set from the other parent. This is why we have two chromosome 1s, one from mom, one from dad, two chromosome 2s, one from mom, one from dad. So what we have is 23 chromosomes from mom and 23 chromosomes from dad. We have a chromosome 1, 2, 3, 4, 5, 6, 7, all of them, 1 through 23 from mom. And then we have a chromosome 1, 2, 3, 4, 5, all the way up to from dad to make 46 total. And so we have 2 of each one, right? You can think of it as like 23 pairs of shoes, right? So the only shoes I have here are my flip-flops, but notice how it's the same flip-flop, right? They're both these reef flip-flops, but they're slightly different. One is for my right, one is for my left foot. So one came from mom, one came from dad. And it's the same for every pair of shoes, every pair of chromosomes. The shoes are different. You have different pairs of shoes. All of these chromosomes are different. They have different genetic information in the DNA of them. But you have two sets, right? You have two versions of each. Haploid, on the other hand, haploid is only one version of each. One chromosome 1, one chromosome 2, one chromosome 3. You don't have two of each one. So it's kind of like if you went through my 23 pairs of shoes and took away one of them. And so for all of your shoes, you only just have one shoe. That's what haploid is. So you have half the number of a diploid because you take all the pairs, you take away one, that ends up being half the total. So if I told you that a somatic, well, we'll do it with humans first. A somatic human cell, so all of my brain cells, skin cells, intestinal cells, they have 46 chromosomes. They are diploid. 46 chromosomes in the nucleus. So they have all the information, all the genetic information. But as we learned after meiosis, sperm cells that are made, as well as egg cells for females, these gametes, right? They're haploid. They only have 23 chromosomes, half of 46. So, '23. And this directly relates. Remember, we get our gametes through meiosis. Well, we were just talking a lot about meiosis, right? How there's 46 chromosomes in the beginning. We start with a diploid cell. But because they line up side by side like this, when I split this, imagine there's 23 pairs. When I split this in half, each new cell is only going to have 23. It's not going to have all the genetic information. So that's sometimes called why meiosis is considered reduction division, because we start with a diploid germline cell is what it's called in meiosis. And then we end up making 4 haploid cells with less chromosomes. There's only one of each homologous pair, one version, because they line up side by side. And then when I split that down the middle, each new cell is going to only have one version. This is different than mitosis, right? Mitosis, we're making identical cells. So if the original cell has all of this 46 chromosomes of information, right? My ending cells better have all 46 sets of information, these chromosomes, because I'm making identical cells. So in metaphase of mitosis, they don't line up side by side. Yeah, they don't line up side by side, but in metaphase of mitosis, they line up single file. So now there's 46 rows here, and every chromosome, because they're identical sister chromatids, all of the genetic information will be retained in each daughter cell in mitosis. But in meiosis here, right, we have 23 pairs. So we only have 23 rows. Here we have 46, because it's one after the other. So really what I'm saying too is that mitosis, you start with a diploid cell, but you also end up with 2 diploid cells. You still end up with all your chromosomes. you just go from single-stranded, right, to, I'm sorry, double-stranded to single-stranded chromosomes, because the sister chromatids separate during mitosis. Yeah. Does that make sense? Questions? Yes, no, maybe so. I feel like that was a pretty good review of a lot of different aspects of, you know, meiosis, mitosis, the different types of cells that do these things. Somatic cells do mitosis, and they're diploid. Germline cells, these are diploid stem cells. They do meiosis to produce gametes, haploid, genetically unique gametes. I think the only thing we haven't talked about is energy enzymes. I talked a little about enzymes with coenzymes. But what kind of conditions will denature an enzyme so that it's non-functional? Temperature, extreme temperature changes, namely hot temperatures will denature an enzyme. So if I ask you, what temperature will denature an enzyme, you'll want to say hot. pH, extreme, so either extreme of pH change, highly basic or highly acidic, will also denature enzyme. Very good, very good. I think those are really the main ones, yeah. But we also talked about a few other conditions, right, or factors that would affect enzyme activity. Substrate concentration, enzyme concentration. What else? Talk about inhibitors. Any questions about that stuff? It's a little more straightforward, I think, than maybe the what we've been talking about, but any questions at all? I think that was awesome. I think we covered a lot of good stuff. Cell cycle, we even covered a bit. We didn't talk much about the electron transport chain. If anyone has questions on that, let me see what subjects. This test is covered, it's unit 3. So if you forget, it's on the syllabus, but it's all, it's going to be energy, additional enzyme information that we covered with energy, cellular respiration, and cell division, mitosis and meiosis. That would be the full extent, and it's 40 multiple choice questions. That's a great question. So the importance of H2O and CO2. Well, CO2 has no role in the electron transport chain. All of the CO2, O2, right. So oxygen, okay. So let's talk about that part. Yeah, exactly. The final electron acceptor, not receptor. Receptors are different, also important in biology. But so the final electron acceptor is oxygen. So that part of the story, let's go over that. So we've got, so the whole purpose, right, of respiration with all of these oxidations, building up NADH molecules and those two FADH2 molecules from the Krebs cycle. It's all for this electron transport chain. And so the electrons from NADH and FADH2 are going to be transferred to that first protein in the chain, and then subsequently will travel down the chain, right? It'll be transferred from one complex to the next. The importance of oxygen as the final acceptor, meaning the electrons go down the chain, one protein to the next, and then at the very end, bind to oxygen. The reason oxygen is important, and the reason why without oxygen there on that last protein before ATP synthase, that's what it's doing, is it's actually pulling these electrons down the chain. Remember, oxygen is this very electronegative molecule, meaning it loves electrons, has a very high attractive force for electrons. It pulls them very strongly. And when oxygen's there, these electrons travel down the chain towards oxygen. Water doesn't really have an important role. All of that's important about water is it's the final end product. One of the final end products of cellular, the electron transport chain, because once the electrons arrive and attach to that oxygen, a more stable molecule has to do with thermodynamics. You're always going to form the more stable molecule in the universe. If it's more stable, it'll happen. Well, oxygen with this electrons attached to it is not stable. It's highly negative. It's not good. So a couple of the protons in that matrix are going to also bind to oxygen to form water. And it's a more stable, one of the most stable molecules you'll ever learn about, water. And it's why we exhale water. But there's no like, it's not doing anything. It's just really the only thing to know is that it's the final, after oxygen picks up those electrons and will bond to hydrogens to form H2O, because it's more stable and it's just the end product. But yeah. That's pretty correct. Yeah. So I would just say the purpose of the electrons from the coenzymes NADH and FADH2. So remember, those are going to be oxidized. The electrons and hydrogen are stripped off, attached to those complexes, and then the electrons moving down the transport chain, right? It is the purpose of them moving down the chain like that, like one at a time, is because if you recall, the electrons, they're very high energy electrons at the very beginning, high energy. Well, when they move to the next complex, it's actually an exergonic transfer. There's energy lost. And we learned from the first law of thermodynamics in the energy lecture that energy can neither be created nor destroyed. It can only be converted from one form to another. You can't, the energy that's lost is actually going to be harnessed by these complexes. These complexes are actually proton pumps. So when the electrons move down, let's say we do one movement down, right? That movement, it goes down, the electrons lose a little bit of energy, they go down in energy, and that energy is used for that first complex to pump that had the electrons. It's going to pump up active transport. Remember, that's an energy requiring process. Pump protons against their concentration gradient, which is something that needs energy if you go from low to high. And so it's building up as the electrons move down. These pumps are proton pumps, pumping up hydrogen ions or protons, same thing, to create that difference. And then, of course, they can flow down through ATP synthase, high to low, during chemiosmosis. Yeah, it's a complicated story, but it all makes sense, right? It just takes some, some careful observation. Yeah, I think we covered everything, or near everything. Any other questions? You're welcome. Good luck, everyone. Thank you, guys. You're going to do stellar. I know it. All right. Thank you, guys. So I'll see you all on Wednesday for Unit 4. Wow, we're almost at the end. Dang. Good luck.